Hello everyone, welcome to Life on Planet Earth. In this chapter, we are going to address evolution on a small scale. Evolution on a small scale, it's basically looking into DNA and looking at the small variations that happen in DNA between individuals in a population. Now, as we go through this chapter, remember that we talked about natural selection in the previous chapter, right? So we are going to also address natural selection here. Now, we say that in natural selection, individuals in a population are going to be genetically diverse, some more suited to thrive and leave behind offspring than others, right? That variation between individuals is what we call microevolution. So evolution at a minute level, right? Now, this picture is that of people who actually live in high mountains. Now, typically, if you living in Ohio leaves Ohio to go to Himalaya, what happens is that by way of adaptation, your body will start producing a large amount of hemoglobin. Why? Because as you go high in altitude, the amount of oxygen in the air decreases. In response to that, your body is going to produce hemoglobin because your kidney is going to release erythropoietin, and that hormone erythropoietin is the one that triggers the differentiation of the stem cells in your red bone marrow into red blood cells. Now, that's the adaptation that you undergo when you go from Ohio, that is not very high, to high mountain. That's what happens. And when this happens, the blood becomes thick. And as the blood becomes thick, the pressure in the blood is going to increase. The heart is going to work very hard, right? And that can cause the heart to give up. So in terms of long-term adaptation, this is not very sustainable, right? Now imagine the people who live in Himalaya and in Montana, in very high mountains. These people actually are native there, so they live there their whole life. They were born there and they're doing everything there. So do they have a high amount of red blood cells so that their blood is thick? Well, believe it or not, no, they don't. What we notice rather, it's that they have a reduced amount of hemoglobin. That reduced amount of hemoglobin is adapted to this particular group of individuals so that they don't have to produce large amount of red blood cells, which would cause their heart to overwork and to give up. Yet, these individuals are just living on top of the Himalaya, just like you live here in Ohio without ever experiencing shortness of breath because you don't have enough oxygen. No, right? They are adapted to live in these conditions. They are suited to live in these conditions. So this is an example of microevolution because these individuals are very much human beings, just like you and your classmates and myself, right? Nevertheless, we are different in terms of how our gene for hemoglobin is different. Let me close that parenthesis. So the question we need to keep in mind as we go through this chapter, it's what is the link between genes, populations, and evolution. Let me repeat that. What is the link between genes, populations, and evolution? How do scientists determine whether a population is evolving or not? Now, remember, it is asking whether the population is evolving or not. Let me say this. Individuals like yourself, you don't evolve. You cannot evolve. Evolution never occurs at individual level. It occurs at a population level. Always, always. All right. So natural selection, it's a set of adaptation, right? That is in response, or rather that is imposed by the environment. The environment is going to cause living organisms, those who have all the features for success in reproduction and fitness to actually leave behind offsprings. So in natural selection, individuals are different one from another, right? Members of a population are different one from another. Two, we can pass down the features from parents to offspring. And then there is an increase in fitness. In other words, those that reproduce are the ones that leave behind the genes for the next generation that are going to be passed down to the next generation, right? Any organism that is not fitted is just going to disappear. Now, selection happens in three kinds. We have a selection that can be stabilizing selection. Another type of selection is directional selection. And finally, you have disruptive selection. What is the difference? Remember, in a population, you have individuals that are genetically different, right? But in some population, over time, as the population passed down genes from parents to offspring, there is a certain set of genes that are preferentially passed down. And those ones are the ones that are going to constitute most of the population at some point. And we are going to see that the middle, the intermediate individuals are the ones that are going to persist over a long period of time. This is called stabilizing selection. On another hand, you have a varieties of individuals, right? Among these varieties of individual, for some reason, one particular group is the one that is going to constitute the major population over time, and shifting more and more toward that group of individuals, so that we have a directional shift like this. This is a directional selection. And then we have a disruptive selection. In a disruptive selection, you have a mix of individuals with different phenotypes. And eventually, over time, we're going to notice that two phenotypes stand out. Everything else is eliminated, right? This is what we call disruptive. So you have one group forming a population here, and another group forming a population here, or now the population evolving into two separate directions, okay? Now, here is an example of stabilizing case. So we have the case of this bird. This bird is producing a number of eggs. We notice that based on individuals in the group of this organism can produce eggs that vary between less than 4 to more than 5, right? So There are less individuals that are going to have less than 4, that are going to lay less than 4 eggs. There are a limited number of individuals that are going to lay more than 5 eggs. The vast majority is going to be right here, laying 4 to 5 eggs, right? What we notice is that in terms of survivorship, the individuals that lay 4 to 5 eggs are the one that are going to be most numerous over time, so that everyone that makes less than 5 eggs is going to disappear. Those that make more than 5 eggs, no, not more than 5, less than 4 eggs are going to disappear. More than 5 are going to disappear, and we're going to have preferentially, after a while, individuals that are mainly producing 4 to 5 eggs. This is a stabilizing selection. Another example of stabilizing selection is that of humans. In the humankind, individuals most likely are going to be born weighing 2.7 to 3.6 kilograms. Okay, now this weight is actually the average. Individuals, babies that are less than 1 kilogram or 1.4 or even 1.8, they have a higher chance of dying. By the same token, babies that are born being heavier than 4.2-ish, right? 4.5, 5 kilograms, 6 kilograms. This is not going to have, these babies are going to die, right? We notice that the likelihood to survive is very high when a baby has a weight that is between around 2.7 and 3.6 kilograms. Okay, that's an example of stabilizing selection. Now let's dive into directional selection. In directional selection, we see that whenever we use antibiotics and insecticide, we are selecting for resistant bacteria to antibiotics and resistant insects to insecticides. So we are shifting from a population that is overall sensitive to a population that is overall resistant. That's directional, right? Another example is the example of this little fish here. This little fish can be very colorful or it can be dull, right? What we notice is that in places where this fish is growing, if there is no predator, this fish is very colorful. So the line that you see up here, the yellow line, is that of the type of color, the number of fishes that occur that are very colorful without predation. Now, whenever there is predation, so in this site right here, where there is high predation, well, individuals are not very colorful. They are going to be relatively drab, okay? So they are drab. They don't attract very much attention, because if they were very colorful, they would be easily eaten, right? Because they would be easily seen, and therefore they would be very much a target, right? This is an example of directional selection. And finally, we have the example of disruptive selection, where you start with a population of snail that is very mixed. But over time, you are going to have snails that are going to be this kind and this kind, so that you're going to have two populations that develop. The intermediate are all gone. okay, over a long period of time. So selection can also be based on reproduction. Now, in the population, whenever there is sexual reproduction, mating is never random. Mating is never random. Organisms are going to go toward the one they feel the more attraction to, right? In the case of these birds here, right, when they spread their feathers, the more beautiful this feather look, the more well-maintained the feather look, the most likely the female is going to be attracted, the most likely the chance of copulation, and the most likely the chance for this particular bird to pass down their genes to the offspring. Okay? So that selection is very intentional, right? That's why organisms tend to have courtship, right? It's very well part of the culture, right? To select preferentially a particular group, organism to mate with, so that the genes can be passed down to offspring. Okay, in natural selection does not always produce perfectly adapted organisms. That is very important to remember. Natural selection sometimes leads to is sometimes constrained by available variations. So basically, what is there? Okay, if there is no particular variation there, well, there is nothing to do to make that variation to be there, right? Okay, so this is going to be the case because sometimes organisms are going to thrive by way of compromise, right? We're going to see that some particular genotypes are not likely to thrive in the environment, while other genotypes have a higher chance of surviving in the environment, and because of basically the environmental conditions. So yes, the alleles are there, and when we say alleles, these are alternate form of the same genes, right? Yes, the genes are there, the variant of the genes are there, but not about any variant of the gene is going to survive. Okay? So, so a population is always genetically variable, right? It's always genetically variable. The genotype, though, that occurs in a population, it's another story, right? Sometimes one particular kind is the one that is going to be favored over any other kind, okay? Natural selection and maintaining variation is ongoing all the time in populations, right? Why? Because mutations always happen, right? Recombinations may happen, right? Independent assortment always happens, right? And fertilization is going to create bunch of combinations in a population. Moreover, you have gene flows that happen. In other words, in a population, you don't only have individuals within that population that are going to be able to reproduce with the individual in the population. Some individuals can come from another population. That's what we call immigration, right? But we can also have emigration, which is that individuals are leaving their population to go in other population, right? This happens across the world where organisms reproduce sexually. You have immigration, you have emigration. Now, these organisms, they don't know anything like they don't know anything like building a border wall, right? They don't know what is a border wall. They just go where they need to go to do what they need to do, period, right? So natural selection is going to favor phenotypes, certain phenotypes, while others are going to not be favored. Some examples of selection where the heterozygous is actually the most prominent and most abundant feature. It's going to be, for instance, the case of sickle cell anemia in populations of African origin. Sickle cell anemia, actually, it's an issue where individuals have a recessive allele twice. So they receive one allele from mom and one allele from dad, and the two alleles are defective. Now, it is shown that in presence of malaria, individuals who are heterozygous who are sickle cell trait are able to withstand malaria compared to individuals who are homozygous recessive, meaning they have sickle cell disease, so their cells in the blood, instead of being round shaped, they're going to be like this, okay? They're going to be like sickled. While the normal shape, it's actually biconcave. The normal shape of red blood cells, it's biconcave, like this, okay? While so this shape right here, it's abnormal. This shape is normal, right? So individuals who actually are heterozygous are able to resist malaria, while individuals who are homozygous recessive, so all their cells are like this, they are going to be sensitive to malaria, while individuals who have exclusively sickle cell, they are going to be very sick, right? So what we notice is that in the population where malaria is endemic, most of the population is going to be heterozygous, okay? So the heterozygous advantage of sickle cell, it's basically that if you are normal, okay, so you have normal hemoglobin, you are very much sensitive to malaria, you have sickle cell anemia, right? You can be very sick due to the sickle cell disease, right? But if you are heterozygous, you are a carrier, you are resistant to malaria, you can withstand malaria. All right, another example of evolution at a very small scale, what we call microevolution, it was observed in the case that we call industrial melanism. In industrial melanism, we see that this moth actually occur in a wide range from white to dark, right? We noticed that at the time when there was not much pollution, the bark of the trees were more clear, right? The white to clear moth were more prominent. But when we saw industrialization and that the barks of trees became dark because of all the smoke, right? What we call the smog, actually, the vast majority of the population went from white to actually dark, that were better able to blend. So we saw a shift in a population from clear guys or white to dark organisms, right? To match their environment, right? So this example is the example of within a population, you have individuals of different kind. Genetically, they are slightly different because they all have a color. But which color stands out depends on which genes which allele you have, right? So we see that in non-polluted area, you have one particular set of alleles that are more expressed, right? Because everything else is going to be eaten by the predators, while in other areas where there is a lot of pollution, some are going to be able to thrive, while the clear one, the white one, are going to be easily seen and be eaten, right? This is another example of microevolution, which is that evolution occurs only at a population level, not at individual level. Now, specifically, changes in allele frequency in a population occurs over time. That change in a population, the frequency of allele in a population over time is called microevolution. If there is something confusing, please let me know. All right, so when we consider a population, that population is going to be genetically diverse. That genetic diversity in a population is called the gene pool. That gene pool can be now characterized. We can choose to look at, okay, white moth versus dark moth, right? So in this particular case, this is the case of peppered moth, right? With capital D representing the dark color, lowercase d representing the light color. So individuals that are dark have two possibilities of genotypes, right? They're going to have a genotype that is capital D, right? Dominant, homozygous. And capital D, lowercase d, heterozygous, right? On the other hand, you have the white individuals, the light color individual are going to be lowercase d, meaning homozygous recessive, right? The frequency of genotype, it's basically the number of time capital D occurs in a population, right? So 0.04 in this study case, while the number of time heterozygous is happening in a population is 0.32 in this particular instant. The number of time light color individuals occur, it's 0.64, knowing that 0.04 plus, so plus 0.32 plus 0.64 equals 1. Okay, it's equals 1, right? The frequency of allele, what is the frequency of allele? When we talk about the frequency of allele, we're going to consider capital D, right? And lowercase d, right? Considering these numbers, what is the frequency of allele of each of the alleles? We know that D occurs here, capital D, but capital D also occurs here. So I'm going to add up 0.04 and 0.16, right? While here we have half of the alleles here are lowercase d, half is lowercase capital D, right? So the half of lowercase d is actually going to be 0.16. 0.16 plus 0.64, that makes it 0.80. So that the frequency of the dominant allele is going to be 0.2. The frequency of the recessive allele is going to be 0.80. Okay? All right. So now let's break down this problem. Okay? So we have the dominant homozygous here, heterozygous here, and recessive homozygous here. So we know that the genotype frequency for the dominant one is 0.04. The heterozygous is 0.32. The homozygous recessive is 0.64. Now, we also determined that the frequency of the allele for the dominant allele is 0.02, I'm sorry, while the frequency of the recessive allele is 0.8, right? So now, while crossing these two individuals, so here we have the eggs, here we have the sperms, okay? Here we have the sperms. So when we cross this sperm and this egg, we have this individual. When we cross this sperm and this egg, then we have this individual. We are going to do the same thing for this sperm. So this sperm with this egg gives us this individual here. And this sperm and this egg gives us this individual here, right? So this is actually called the Punnett square. Okay, the Punnett square. We are going to see this a little bit more when we looked at genetics, right? But to come back here, in terms of population genetics, the genotype frequency, I already showed this earlier, 0.04 capital D plus 0.32 capital D lowercase d plus 0.64 lowercase d all together equals 1, right? Now, capital D is can also be written p square, while lowercase d, it can also be written q square, while the heterozygous can be written 2pq. So 2 times p times q. Now, considering the values here, this would be right here, this is p and this is q. Likewise, this we have p and this we have q here. Okay, so knowing all of this, now we can run our numbers. P square, it's basically 0.2 times 0.2. So this one times this one, this is what gives us this, right? 0.04, right? Likewise, when we look at this 2 right here, okay? So we have 2pq, 2 times 0.2 times 0.8, all together, it's 0.32. Finally, q square, it's 0.8 times 0.8. So that's 0.8 square. That's 0.64, right? All together, it's 1 here, okay? So please practice this, okay? Practice this to understand the relationship between genotypes and phenotypes, right? Now, I'm not going to bug you too much with that at the exam, but to some extent I will, right? So please practice this. Okay, so there were 2 guys Their names were Hardy for one, Weinberg for another. These two guys actually simplified the way we look at microevolution, right? How did they simplify that? They said, well, we are going to calculate in a population the frequency of alleles, right? Because you have a variation in genotypes in a population, we are going to find exactly, right, what is the proportion of a particular genetic variation in a population. And by looking at that genetic variation, if we see a shift over time, we are going to say that microevolution is at play. Let me repeat that. When you consider a population that is genetically variable, right? When do we say that we have a stabilizing selection, a directional selection, or a disruptive selection? Well, we can say that if we see a shift in occurrence of alleles in these populations, right? And Hardy and Weinberg say that in order to be able to come up with such prediction, we have to assume that there are not many disturbances in the population. No one is coming in, no one is going out. There is no mutation, there is no random mating, right? We always have to be able to predict how the mating is going to happen, right? And there is no selection, right? But we know that in a normal environment, that doesn't exist, right? But for simplicity and to understand microevolution, we are going to pretend that the situation is ideal, right? And we only have two types of allele in a population, a dominant one and a recessive one, right? A dominant allele is always P. A recessive allele is always Q, right? So that P is the frequency of the dominant allele, while Q is the frequency of the recessive allele, because each individual receives one gene from mom, one gene from dad, one allele from mom, one allele from dad. So that individual is one single individual. Therefore, one allele from mom, one allele from dad, that makes it one individual, right? So P plus Q equals 1. But now, let's take a look at the genotype. So here we were taking a look at the frequency of allele. Now let's take a look at the frequency of genotype. So the genotype are going to be here, P squared, that would be, for instance, in the previous case, capital D, that's a genotype for this individual. Q squared, it's lowercase d, right? From the previous case study. And finally, heterozygous 2pq is going to be capital D, lowercase d. I want you to remember that, okay? And this is nothing but the genotypes, okay? So now, Hardy-Weinberg said, well, there is no mutation, there is no gene flow, there is no random mating, there is no genetic drift, there is no selection. That's the assumption of the equilibrium in a population based on the Hardy-Weinberg principle, right? There is no modification that ever happened in a population, right? This is the case right here. But we know that in real life, that doesn't happen, right? So Hardy-Weinberg principle condition rarely ever occur, right? So allele frequencies are going to change in the population because there are individuals that come in and individuals that leave the population, always. Therefore, microevolution is always occurring, right? So, but nevertheless, to study microevolution, it's very important to keep into account the Hardy-Weinberg principle, right? So, what causes microevolution? Microevolution is caused by genetic mutations. If you see this individual right here with freckles, right? One can wonder, well, if freckle is dominant, why is that most of the population doesn't have freckles? Well, it just depends on when mutations are happening. And what is a mutation? A mutation is when the sequence of DNA changes because of some accident, right? So here we have a normal sequence that is G, T, G, C, but because of radiation, that C can be changed into A. Now the new sequence is saying G, A, G, C. One single mutation, right? This one single mutation can be enough, for instance, for someone to be lactose intolerant, right? While everyone else is digesting lactose, well, that one single person with the mutation is going to be lactose intolerant, okay? All right. Also, we do have a source of microevolution, gene flow. Now, we see the distribution of these rat snakes in the United States, but these rat snakes do not occur in one single color. Even though you have some that are more prominent in one area compared to other areas, everything depends on where they can camouflage well, right? When they can thrive and leave behind offsprings. But guess what? When they come together, they get to copulate and produce living organisms that are going to show variant of a color, right? And this is because of non-random mating, right? They show preference, but at the same time, they are able to reproduce among themselves because some are going to leave Florida to go toward the upper part of the US. Others are going to leave the upper part of the US to come down here and be able to reproduce with other organisms that way, and so that we're going to have a wide diversity of these rat snakes. Okay, another reason for microevolution is the genetic drift. Say we have a population here. That population is made up of a number of frogs with different phenotypes. Now, a catastrophe can happen. For instance, a flood, a very cold winter, an extremely hot summer, right? Some disturbance in the environment so that this few guys find themselves isolated from this big group. As consequence of that, this individual that only represented 10% of the population in here is now going to represent 20% of the population in here, so that this individual here has now more chances to actually multiply and leave behind offspring, so that the frequency of this allele is going to increase, right? And this is going to constitute a shift. And finally, but not least, cause of microevolution is the bottleneck effect. In a bottleneck effect, you have a population that is very genetically diverse, right? And that population that is genetically diverse, for some reason, something happens, right? So that only few are selected. And the ones that are selected are the ones that are going to make up the new population. This is particularly true in the case of founder's effect. Now, when Europeans started leaving Europe to come and populate the United States, well, they didn't choose specifically what individuals with which particular genetic background would come in the United States. No, it was completely random, right? People just felt that, okay, this area is crowded. I'm not thriving here. I'm going somewhere else. I'm going in Americas, in the New World. So when they came there, they came with their genetic background. And as a consequence of that, when they came, they reproduced among themselves and caused the same sets of genes to be present in a population, and the proportion of these genes obviously became very high compared to their population of origin in Europe, right? So this is the founder's effect. Now, when it comes to human population and other animals, the founder's effect, it's not always favorable because we see a lot of genetic aberrations, abnormalities that are happening in a population, right? That's why in the human culture, we say, well, first cousin should never reproduce together, right? Because chance that they're going to pass down accumulations of mistakes in the genes is very high. This is where we are going to stop this presentation. But before we say goodbye, please review your notes. Go at the end of the chapter in your textbook and review all the content. Design some questions. Answer some questions at the end of the chapter so that you can practice your understanding of the material. And finally, but not least, create a handwriting concept map. It's going to be very helpful to grasp this material. Thank you for your attention. Bye now.